All, welcome to one more show. This is Dr. Mubin Sayed from drbeen.com. So, over the weekend, I had been looking at these news that Omicron may have picked up a gene from a common cold virus, and that may have been the reason that it has become more transmissible. And it was interesting. And I also thought that as much as we are thinking it is a um, less mild variant, there must be a thought in our mind as well that is it picking up genes from other viruses and what is going on. So, right in the morning, I haven't yet gone out for coffee. I was actually preparing. This needed a little more preparation and reading. So, I've been doing that since the morning, and here I am. So, let's start our discussion. I'm going to do two things. Number one, I'm going to go through the paper, and then I would explain the mechanism in my drawings to see how do they think that this is happening. So this is drbeen.com. As usual, references. This is the reference to the company. nFerence is the company that have made this study. This is another study that is interesting. That is the template switching. And this term will come up in a second and I'll talk about it. These are some news that you might see. For example, Omicron COVID variant may have picked up a piece of genetic material. Omicron variant may have picked up a piece of common cold. Omicron variant may have picked up common cold virus. Omicron possibly more infectious because it shares genetic material and so on. And then here, if you see, if I search for Omicron more transmissible because of common cold gene, and then you see a bunch of the Hill is here, the ADN is here, routers is here, and so on. The point is that you would hear this more and more, and you'll see this more and more. And I wanted us to be in depth aware of what is going on and be able to think about it clearly. Then this is another study that is important. SARS-CoV-2 spike protein, key target for eliciting persistent neutralizing antibodies. So more important is the spike protein genetic material to be able to see during a talk. This is another reference for the genetic material. And then this is a reference for the other human coronavirus gene that we would talk about. So that is the HCOV229E. So these are the references. Now let's look at this study, what they're saying. What they're saying is the following. They're saying that Omicron is clearly more transmissible. Good. We all at least know that much from the data so far. Secondly, they're saying there are 26 amino acid changes in the Omicron's spike protein. These are one of the largest changes so far. And here they say 23 substitutions. So there are 23 amino acids which have become changed with other amino acids. 2 deletions, 2 amino acids have become deleted, and 1 insertion, and 1 amino acid has become inserted. So they're saying that these are distinct compared to many other variants. And what they did was, the researchers here, they picked up alpha, beta, delta, other variants, more popular variants, more transmissible variants compared to their ancestors, and they looked at their genomic sequences and they compared them to Omicron. The thing that they say they are, what they were trying to figure out is what are the common variations that are common to deltas and betas and alphas and to Omicron. And then they are kind of assigning those to not be the reason for the higher transmissibility of Omicron because they were present before as well. However, then they were trying to see what are the newer variations that are just unique to this variant. And in that, they found this one variation, and I would discuss that. That variation is an insertion at the 214 position in the spike protein, and the insertion is EPE, and I'll discuss what that insertion is. And they're saying that this one code sequence, amino acid sequence, is also present in human coronavirus 229. Number one. Number two, they're saying that in our GIT, human GIT, and in our respiratory system, the cells have receptors for both coronaviruses, human coronavirus 229 and SARS-CoV-2 or Omicron. And because of that, both of those viruses can simultaneously infect a single cell. And of course, that would increase, the chances of doing that would increase during the common cold seasons as well. So when the human cell is co-infected with two viruses, then uh, the authors say, the researchers say, that it is possible that SARS-CoV-2 may have, the Omicron may have picked up a genetic piece from the genetic pieces of human coronavirus. And that genetic piece is the reason for possible more transmit, one of the reasons. There are other mutations too. The other thing that they said was that this genetic piece, this mutation 214 EP or EPE sequence of amino acids, is also seen in human genome. So our messenger RNA would also have EPE in it. And they said, so the only possibility for the virus to pick that from somewhere will be either from the human genome or from um, the human coronavirus. I think that there could be mutation as well, a spontaneous mutation too, that could do it. But they feel that that is the possible mechanism. So here we stop. The reason that they say this may be the part of the more transmission is because they feel that this change is in an area where the T cell receptors bind for killing the virus. And they're saying it is possible that that binding is not working correctly as a result of this mutation. So T cells are not able to activate the immune system very well. And that is why the response or the evasion is occurring. I would actually possibly think, if that is happening, if then maybe that is the reason immune system is responding lesser in intensity, and cytokine storms may not be occurring that frequently because T cells are not becoming active as much as they would with the other variants, because they're not able to bind and figure out every single time there is a virus. So that is the general discussion of this. Now let's go over some of the illustrations, and I would then go over some of the paper itself as well. I have actually, if you can see, here is a paper. So if you just wanted to hear the summary for what they're saying, this is a summary. You can, this is in a few minutes done. Now let's look at the details of it. This is the paper, and I would discuss that paper now. And here are my illustrations. So here we are with our coffee shop for humanity and the gifts in there. It is actually funny that nowadays when I go to Starbucks or other places, there are actually gifts placed there as well in the coffee shop. So gifts for humanity and humanity's coffee shop, we are here. Now, very simple. We know this much, that we have for the SARS-CoV-2, we have spike protein with S2 unit, subunit with S1 subunit with RBD on it, and with the N-terminal domain. N-terminal domain is usually the beginning of something. So for example, if you look at this little pen in my hand, and let's say that this side, the one that my finger is tapping, this is the beginning of the pen and this is the end of the pen. That is the same thing for the RNA or the nuclear material as well. We say there is a beginning side and there is an end side, and the beginning side is usually 5 prime and the end side is 3 prime, and in the positive sense, or it is usually the N terminal domain for an amino acid or a protein. So N terminal domain simply means beginning of something. So for example, if I look at this genetic sequence here, let's say this is the genetic sequence for the spike protein. It starts from here. The beginning of the spike protein area will be called N-terminal domain. So this is important to understand their paper, that they are saying, we know that the N-terminal domain, this whole part is actually part of receptor binding domain. I have made the N-terminal domain a little farther, but this whole thing, imagine, is receptor binding domain. This is the part from which the virus connects with the cell. In the N-terminal domain, the early part is the super antigen part or super antibody part. That means any antibody that binds here usually is able to help neutralize the virus. Fortunately, this change, this insertion of a new set of amino acids, is not in this N-terminal domain at the super antibody part. It is a little distal to it. That is a little after that. And here is that change. There are three amino acids added, E, P, and E is a sequence. And I'll explain what these are. These three are present in an area of the spike protein where usually, this is still in the early part and terminal domain, it is still on the RBD. This is where usually T cell binds or T cell receptor binds. Of course, T cells also need receptors to bind, right? So this is a T cell, and it would bind here to start getting activated. And then it would become activated with macrophage, and then it would activate the remaining immune system, the B cells or the T cell. This initial binding is important. And what they're saying is that it is possible that this insertion of 214 EPE is kind of defeating or reducing the capability of T cell to bind here, which would in turn make the immune system's response a little lesser. This is what they think. And again, they're not claiming that this is exactly what's happening, but this is a conjecture. Now, how is it happening? Going a little more deep in this. And before we go actually there, I want to show you, my apologies, I'm so excited in the morning to discuss this so that you don't become more concerned. So here is the spike protein genes. And if you see here, this is the beginning part, and that is the N-terminal domain. This whole part here is the S1 subunit. Within the S1 subunit, this is the receptor binding domain area as well. This is the receptor binding domain. This is N-terminal. So this changes somewhere over here, kind of outside of the receptor binding domain. So I tried to correct it to the receptor binding domain. This may not be exactly correct. But anyways, N-terminal domain. It's difficult to make 2D and 3D together. Now, another thing, there is one more diagram. This diagram is also good. So here, this is the single-stranded genome. So keep this in mind as well. In this diagram, please look at this 5' over here, because we're going to talk about this in a second. A virus, these coronaviruses, are mostly called positive sense RNAs. And we have done this discussion 2 years ago. Unfortunately, it is 2 years in the pandemic. 5' is the beginning of the RNA, and 3' is the end of the RNA. And this is called a positive sense. In negative sense, we'll have 3 in the beginning and 5 at the end. So, and I have done a detailed discussion, 1 hour long talk about it. So I'm not going to go in the further detail today, but just keep this in mind as well. And now I'm going to go to explain and keep this one as well, HCOV 229E. So this membrane-bound zinc metalloprotease, this enzyme, ANPEP, this is another receptor we're going to talk about. This is the receptor that is for HCOV 229. So now let's look at what is going on. It's actually very simple, but when you read it in text, it sometimes can look a little daunting. So here is, let's say, Omicron or SARS-CoV-2. Let's say Omicron's father or mother. And here, or father, mother together, someone. And here is the humanized coronavirus 229, right? So human coronavirus is binding to its own receptor, that is ANPEP. This receptor is found in the cells in the GIT of ours and respiratory system. And we know that the SARS-CoV-2 binds with ACE2. And we also know that when SARS-CoV-2 binds to ACE2, and this is the same thing for the other side as well, the HCOV, human coronavirus, when they enter our cells, then they would shed the protein, the messenger RNA, positive sense RNA would come out. Positive sense single-stranded RNA would come out. This RNA would then work with the ribosome to start making its own genetic material. One part of that is to make RDRP, RNA-dependent RNA protease. And I'm going to talk about that for a second. So let's just look at that. This is the last part, and then we stop our talk. What happens is, when the virus brings in its genetic material in our cell, imagine you are looking now inside the cell, outside of the nucleus, inside the cytoplasm. You're seeing there, this is rough endoplasmic reticulum. This is the Golgi reticulum, smooth reticulum with the ribosomes on it. So for the medical professionals or students, just to give you the location, for us all in general, cytoplasm outside the nucleus. So here the genetic material has a 5' end and a 3' end. And this is a positive sense single stranded messenger RNA from the virus. Good. Now, to make new viruses, we have to make copies of this. To make new viruses, we have to make copies of this. So let's say this thing needs to be made in multiple copies. So multiple viruses can be made, and every one of them can be given this tiny brain. So that's one. Second, various parts of this RNA, you know that genetic material has spike protein on it and open reading frames on it and so on. Various parts of this RNA actually make various virus enzymes. Those enzymes are responsible to work for the virus to build more viruses. So this virus has the responsibility that not only it has to make more copies of its RNA, let's call that template RNA, or the original genomic RNA, the ancestral RNA, the parent RNA that came in. All these terms for the same RNA that came in. We have to make more genomic or ancestral RNAs to put them in the new cell, in the new virus, right? So that is point A that we need to accomplish, the virus needs to accomplish. The second is it needs to make its enzymes in this cell so they can help with the building. It needs to make its workers. And how does it do that? It is such a clever mechanism, and all viruses do. What it does is it would take pieces of its genetic material, make small genetic pieces from that. These are called subgenomic RNAs, parts of RNAs. Imagine you take a brain of something and take pieces of the brain and let those pieces do some function. And from these pieces, we make enzymes. So that means the virus has to accomplish another task B, and that is to pick up its parent's genetic material or its own genetic material, because it is a parent which is doing this all at the moment. Then from those tiny genetic pieces, extract the tiny genetic pieces and then make enzymes from there who would become workers, and these workers would then help inside the cell to promote further duplication of the virus. This worker should look happy, right? That is a task B it has to do. Now is the important thing. But to do all of this, the single-stranded RNA has to be first copied in a negative sense. So from positive sense, it has to be copied to negative sense. Then when the copy of that is made, then these would be positive sense. Why is this? The problem is that our copying mechanism in the cell, we call them polymerases, RNA polymerases. This is actually the virus's own enzyme, RNA polymerase. When it copies the genetic material, it actually reverses the base order. This is like if you look at yourself in the mirror, you are actually reversed, at least horizontally reversed, not vertically. Correct? You are reversed. Then if you look at the mirror in the mirror, then you are once again corrected. So this is what happens when the first time RNA polymerase acts and tries to make a copy of this positive sense. It actually ends up with an inverted copy. Then it makes another copy of the inverted copy, and that would produce the correct copy. Right? So this is what has to be done. 2 steps. You go from positive sense to negative sense back to positive sense. Number 1. Number 2, we also create the subgenomic RNAs. That means now this is the key discussion part. That means whoever is helping make the copy might make the copy, start making the copy. Imagine you are the one who is making the copy of a long thread. In the middle, at some point, you might disengage from making this copy because you just want to make a subgenomic RNA. and that small piece that you made is sufficient. Or you might disengage and then re-engage at the same place, thinking, oh, I think I should just continue to make the main brain, ancestral brain. And so there is a possibility of disengaging and re-engaging, or disengaging and then re-engaging somewhere else. And in this disengaging and re-engaging, there can be an error made where another piece of RNA that was just roaming around doing its own fun things from other HCO or human genome might get inserted in there, might get accidentally caught and become inserted in there. This is like somebody's in a chicken, what is chicken farm, and trying to catch a chicken, and one chicken runs away and he picks up the other one by mistake and starts using that. So let me now explain that part. So the RDRP, RNA dependent RNA polymerase, is an enzyme that helps copy the genetic material of the virus to make more genetic material copies. So keep that in mind. This is virus's enzyme. This is a different discussion, that how did this enzyme come to be. We can do that discussion at another time. I've done that in the previous talks. So here, this is our RDRP. Check out some things here. It is connected with original RNA that came in with the virus. So this is the insertion of the original RNA. We also call it template RNA because we read this, or our RDRP reads this and makes a copy of it. So this is the template and we'll make a copy. Here, if you see in this little black thing, this is the copied RNA coming out of the RDRP. So RDRP goes over the thread, and as it is reading it, it is copying it as well, and a copy is coming out. Now, we also know that the original is positive sense, but the copy that is going to come out is going to be negative sense. RDRP would have to then engage with the copy to make a copy of the copy, and that would make the original like. That's the general process. But what I want to discuss is something else here. RDRP can, in this process, disengage. This is called template switching mechanism and switching by choice mechanism. So let's look at what is happening. So RDRP disengages, and here you can see that RDRP is present and it has disengaged from the template thread. So whatever is the genetic material produced is the genetic material produced. It may not be complete. One possibility. Second possibility, RDRP re-engages at the same place where it disengaged and just continues on its merry way from there, and what comes out is normal copy. That's second possibility. Third possibility is, if you see in this one, RDRP disengages from the template that it is making copy of and then re-engages slightly at a different position. And now we would have this copy that would come out is going to be different. This is equal to, if you're making photocopy of a book, you take half page copy from the upper part of a page, and then the lower part you take a copy from some other part of the book. So now you've made a Frankenstein page, which upper part is different and lower part is different. So that is possible here, third choice. A fourth choice, this is important, is that RDRP can actually, when it is disengaged and wants to re-engage, it accidentally or by choice picks up a different RNA, which is homologous or homologous, I guess that's how to pronounce it, which means similar looking. So SARS-CoV-2's genetic material and human coronavirus 229 would have very similar looking genetic material. So it is possible to accidentally pick that up and just start working on that, thinking this was the original. Right? So this is like trying to catch one chicken, and instead of that, you accidentally catch another one. So this is the possibility where now some part of that new genetic material template that we got, this is template switching that has occurred, some part of that get inserted. Another possibility is a heterologous genetic material is picked up. That means a genetic material that is not even like this one, the original one. So instead of picking up the genetic material of human coronavirus, it picked up humans' genetic material, our RNA, and then used that to insert something. So that means when these two viruses co-infect in GIT or respiratory system, it is possible that the RDRP that is making new brains might end up creating a template switched brain with some insertions from a different RNA. And that is their thought. Now, how much that is possible, how how good or bad that is, we'll see. But this is what they've done, and I want to show you their diagram, which is also interesting. So if you see here, those who are once again more interested in what exactly happened in this insertion. So here on this red side is the human coronavirus 229E. Let me see if I can make it a little more big. This red side is human coronavirus, and that is also infecting the same cell and it's doing its own replication. You would see that the replication is top is positive sense. Then in this gray box, the positive sense converts to negative sense, then converts back to positive sense, as we discussed before. So here, both viruses are replicating their DNA. Their RDRPs are working. And now this virus has converted from RNA positive to anti-RNA or sense, so the negative sense. So if you see, the 5 is here and now 3 is here. Now, while this is happening, on this side, the SARS-CoV-2 is also doing its replication, and its RDRP has changed from 5 prime to 3 prime and disengaged. And when it re-engaged, it picked up human coronavirus's sequence and it added that into this one, into SARS-CoV-2. And the end result was, if you see here, glutamate proline glutamate, or EPE, is formed. And the amino acids GAG, CCA, GAA, these are the codes, nucleotide codes that are going to make glutamate pro and glutamate. So that is what is happening. That is insertion 214 EPE. And they have more discussions if you wanted to go more um, more in detail. But I would stop here for the discussion. So Eureka Hope says, I was trying to be respectful and understanding, but overzealous deletions are reminiscent of the lies those in power in the beginning of this pandemic. Power corrupts. Okay. All right. So I wanted to make sure that you are aware of this right in the morning. You can do your own reading of this paper as well if you think that things would be different. At least you'll be able to see what that paper is. So far, again, it looks like it is good. How about we do this? I'm going to disengage and we'll meet again at 6 o'clock. I saw a bunch of news that is interesting as well. If you want me to do a news roundup, I can do that in the middle of the day. But thank you very much for joining in the morning, bright and early. Love you all. Stay safe and happy and healthy. If you would like to support this work, there are links in the description. You can buy me a coffee, or you can use PayPal, or you can become a patron. And the easiest help you can do also is that you can just like, subscribe, and share. Thank you very much, and I would see you later today.